afternoon to everyone. Buenas tardes to all. It is my pleasure to welcome you back to our BP Endure virtual summer series. And today we have the opportunity to be with one of our BP Endure scientists, a part of our network and of our community, Dr. Hamilton Farris. Dr. Farris' career involves three aspects of science education at LSU, Louisiana State University School of Medicine: mentoring, teaching, and research. As an assistant dean of student affairs, he advises MD and MD PhD students on academic, disciplinary, student health, career, research, and extracurricular issues. Imagine all that plus being a scientist. With regards to teaching, Dr. Farris is co-director of the neuropsych training for preclinical medical students. But in his lab, he integrates neurophysiology, pharmacology, evolutionary and developmental biology, and psychophysics to elucidate the mechanisms of sensory processing. In fact, Dr. Farris loves the auditory system. And in his lab, his members take a neuroethological approach, determining how the nervous system and behavior functions in their natural context. And most of their work focuses on the mechanisms and evolution of auditory and visual sensitivity in non-human animals, with publications in high-impact journals like Nature Medicine, Nature Communications, and Science. And Dr. Farris received his PhD from Cornell University and his bachelor's in science from the University of Mississippi. And one fun fact is that he wants to come to Puerto Rico to listen to coquis because he works with frogs. So it will be really wonderful. Once COVID allows traveling safely, we would love to have Dr. Farris come and visit us and continue this conversation of this work that he's doing. So without further ado, Dr. Farris, thank you for being here.
Oh, thank you very much. Should I share the screen right now?
Yes, thank you.
Okay. Can you all see that?
Yes, we can.
Make sure. Is it in slides for you?
It's not in slideshow yet, no. But we can see it without a problem if you have
one second.
Okay.
There we go.
Yeah.
Okay, sorry about that.
Yeah.
It's a real pleasure to be here, and I really appreciate the invitation. This is a really important program. I just want to say from the start, this is a tough time we're living in, and if this had been 5 years ago, we wouldn't have been able to do this. And so with the glass half full kind of view, we should take some encouragement that we're able to keep some momentum going with our education and our research and our teaching. And so these kind of opportunities are very meaningful to me, and I'm excited to talk about some of the work that I do with you. I'm an interrupter, so if you'd like to interrupt at any time to ask a question, I'm happy to take it, or you can save it till the end. Just a disclosure, no commercial interest in this talk today. So I'm going to talk about sexual dimorphism in vision, and I'm really going to spend some time. Almost all the data you'll see today were collected by students at your stage right now, which is really exciting. You think that you get into education and you have to do so much apprenticeship and so much practice working in a lab or taking classes that you have to wait to make an impact with scholarship and knowledge production. By the way, that's what a PhD is. It's a knowledge producer, taking things from the unknown and making them known. And the work that I'm going to show you today, the data were all collected by four either undergraduate or postgraduates. I'll highlight them: Whitney Walkowski, Caitlin Leslie, Robert Rosencrantz and Keith Perkins. So let me talk about frog eyes and vision for a second. And I'll tell you what my motivation is. So if you were an actor, and you said, what's my motivation? Well, my motivation as a scientist, I'm really interested in how sensory systems work. In fact, a really complicated way that they work is that we integrate different sensory systems to make a single perceptual outcome. And one of those integrations is between the auditory system and the visual system. And I'm showing you the animal that I work on the most. It's called the tunguro frog, and here's a male floating and singing right here. And when it sings, and I'll play you the call, and I'll show you a video of him singing in a bit. When it sings, it inflates its throat sac as it's singing, and it's also producing a sound, the call, that functions as a sexual signal that females are using to make mate choice decisions. And so in order to process these two signals, the sound will go into the ear and into the eighth nerve, into the brainstem. So this is the spinal cord here, brainstem. This is the forebrain up here. You'll have this processing of auditory information ascending in the nervous system. But also you'll have a visual input, which will go through the eyes, into the retina, into the optic nerve, and then into the midbrain right here. And so there's audio-visual integration in the midbrain. And I really would love to understand this complicated question of how you put together in time and in space an acoustic stimulus, the sound, and the visual stimulus, which is the light reflecting off the moving throat sac. So let me show you some examples of this integration. Now, I can't play this out of PowerPoint. We found out yesterday, so I have to unshare this and show you and play it through Chrome, but I'll do it in a second. I'm going to show you a video of a person talking, and then I'll show you a video of the tunguro frog singing. So bear with me one second. Okay, can everybody see this guy here, this video? Yeah? Yes. Good. All right, well, what I want you to do is I want you to really look at him and listen to what he's saying. So everyone look at his lips primarily as I play the video. And I'll play this several times, but look at his lips. Okay, one more time, looking at his lips. Ready? Now, I expect that most of you heard da's, as in D-A, da, 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 da. You might have heard a ga in there, a G-A. Now close your eyes and just listen to the audio. Don't look at it, close your eyes. Most of you have now heard the phoneme ba, B-A, which you didn't hear before. And I'll explain to you what the audio and video are doing right there, because clearly there's a difference between what you hear when you're looking at him versus what you hear when your eyes are closed. So now let me play for you the tunguro frog singing. So this is a male in a puddle, and this is in Gamboa, Panama, where I work most summers, unfortunately not this summer. And he's calling to attract a female. It's at night, but the light from the camera has illuminated him. So here's a male singing. Okay, so you can see that there's a coupling between the sound and the movement of the throat sac, just like there's a coupling between the sound and the movement of our lips when a human speaks. So I'll go back to the slides now. One second. There we go. Oops. Okay. So when you were listening to this and watching it, you probably heard da da, as I mentioned. The audio had bars in it, which you couldn't perceive because the lips were actually making the ga movement. And when you combine the audio and the visual together, the percept is da 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 da. This is called the Mcgurk Effect. It's a famous effect, and it illuminates in a behavioral test that, for sure, the auditory processing in your brain is interacting with the visual processing. That means that these are not separate systems. You often learn them as being separate in a neuroscience class, but eventually they come together to form some common percept. For a female walking along the forest floor looking for a male, she's seeing this when a male is about to sing, and then when he produces the call, she sees this movement here. And there's plenty of research to show that females are integrating, just like we do, the auditory and visual information to get this third percept of a combined auditory visual stimulus. So I'm interested in how that information gets together in the midbrain of these frogs. And by the way, the anatomy of their midbrain is quite the same as our midbrain anatomy as well. So when we started down this project, started down this road looking at this question, the first thing as neuroethologists, we needed to remember that we need to look at the nervous system in its natural context whenever you are exploring an animal model. And we're looking at an animal that's integrating sensory stimulation when they're undergoing reproductive communication, meaning when they're mating. That means that their brain and their nervous system is in a different endocrine state, meaning a hormonal state, than when they're not mating. And so this raised the question that we need to look at how the sensory systems work before we get really into this multi-sensory integration. We just need to know what's happening to the visual and auditory processing when the endocrine system is turned on for mating behavior. And any of you have studied things like ovulation, implantation, and pregnancy, gestation. You know that we undergo this huge change, humans that is, this huge change in endocrine state when we're undergoing a reproductive event or preparing for one. And it's the same endocrine modulation that you see in other birds as well. So before we can get to a sensory integration, we start with understanding sensitivity in the neuroperiphery, meaning the eyes and ears, under mating conditions. And that's where I'm going to focus today. I'm going to talk about how endocrine changes, meaning hormonal changes, change processing in the retina. So I'm not going to talk about auditory information, unfortunately. I mentioned before, it's my favorite sensory system. But I'm working on the visual system right now. It comes in a close second. It's not so far behind. So what does the frog's eye tell the frog's brain when evaluating a mating communication signal? We need to put the frog in the correct endocrine state and see if that changes the neural processing of the retina. What's my second motivation in all this? So you can obviously see I really love the biology of these animals, but my second motivation is that we're diurnal animals. We are day-living animals, and so vision is critical to our behavior. Vision, if you are blind, if someone is blind, it really does limit the types of things they can do, and they have to compensate in a number of different ways, but nevertheless, it presents a significant handicap. There are about 45 million people in the world are affected by some form of severe blindness, and the blindness can be in forms of optical blindness, which means that you somehow prevent light from passing through the cornea and lens and vitreous to get to the retina. So you can have a parasitic effect inside the vitreous, like you see in cases of river blindness caused by nematode. You can have optical problems where you've misshapen the lens or the shape of the eye and cause blurring, like I do and many of us do. You can have a cataracts in which you block the light from passing. You can shade the light from passing through the lens. And these are all optical forms of blindness, but also more devastating, if you will, because there are really no therapies for it, is the loss of the neuroretina. So this is neural blindness. And this can come from glaucoma or causing macular degeneration, in which there's a change in blood flow and fluid exchange in the globe, and so that kills the retina. You can see what these pictures are. What these pictures are, you're looking through the lens into the back of the eye. They're called a funduscopic exam or image, and fundus means the base, like fundamental. It means the base and the cup of the eye, and that's what an ophthalmologist does when they look through very closely with ophthalmoscope. And so a normal eye should look like this. You should see the blood vessels heading back through the disc. You should see this nice pink area in the back. That's the retina. If it falls apart, it's going to look like this. If it falls apart in little pieces, it's called retinitis pigmentosa, which means that the retina looks pigmented. And this is the back of the eye, the retinal pigmented epithelial cells showing through for what once was nice pink neural tissue. And so this is loss of sensory space. This is like removing any receptors for light in these parts of the retina. And so this person has less ability to see with this eye, so too with this eye. So the question I have in this part of the motivation is, can we come up with a therapy, or at least a tool for enhancing the sensitivity of an eye? It could be a drug, it could be any type of thing, but we're thinking about a therapeutic approach. And so there's a biomedical goal to this research too, and that's to establish an animal model for modulating sensitivity in a retina. In this case, I'm showing in a remaining retina. So I'm going to give you the punchline first of this talk. I'm going to show that endocrine, meaning hormonal modulation of the retina, exists and that it causes a change in sensitivity. And this conclusion I'm giving you right off the bat is built on 3 lines of evidence. The first is from the literature, not my work, is that retinal integrity is endocrine dependent. So things like hormones like progesterone initiates pro-survival effects of the cells in the retina. There are also neuroendocrine circuits in the retina. Just histology has been done to show that we've got hormonal receptors, primarily estrogen receptors throughout the cell layers of the retina, suggesting that a circuit exists, a signaling pathway or circuit, however you want to look at it, exists for modulating the function or even the homeostasis at the very least, but modulating the function of the retina. And then our discovery that these students made is going to show you that we can shift the sensitivity of light to a more sensitive retina using a form of luteinizing hormone, which is called human chorionic gonadotropin. So this is what we're going to talk about today. So I mentioned sensitivity to light. What determines sensitivity of an eye? There are 2 types of sensitivity you can measure. The first is called the optical sensitivity. It's really just based on the morphology or anatomy of the eye. So there's an equation for this called the Land equation. This S means sensitivity. It basically takes several metrics of the size of an eye. And it's basically treating the eye as if it's a camera. And it's asking, how big are the structures in it that are collecting light? So if you see this A right here, that's the aperture. So this is an eye we're looking from the side, by the way. Here's the globe, there's the lens, there's the cornea. So this is outside, this is heading back to the brain. So here's the lens right here in a frog eye that's been sectioned. And we're looking at the aperture, how big that is, the focal distance, how far back to the retina. So the retina is running back right there. And then how much light can be captured by the photoreceptors. So you have a photoreceptor here, so light's going to come in through the lens at the aperture. Here's the focal distance, and at the back, you have a photoreceptor like this rod, which has an outer segment that captures light. I'm not going to go into the phototransduction mechanisms today, but suffices to say, the longer this outer segment is and the wider the diameter, the more light it can capture. If you put those into an equation, you end up with a sensitivity that means that comes in these units, which is the amount. It's called steradians by microns, by square microns, and it just means how much of the visual field can be focused on a single receptor. So how much light can be put into a single receptor? So there's not a single sensitivity It's not a single sensitivity because in all eyes, because species live in the daytime and the nighttime, and there's a 1,000-fold difference in the amount of light that's out there. So if you go looking for one single sensitivity, you're not going to find it. You have to look across species to see how light sensitivity changes based on eye morphology. And so for today's talk, I'm going to primarily, in the first part, I'm going to talk about 4 species, 2 that are nocturnal. They live at night, they're out at night, and they sing and attract mates at night. So they're using their eyes primarily at night. And then 2 that are diurnal, meaning they're out during the day. These are the so-called poison dart frogs, which are really beautifully colored. And they have all this nice coloration on their backs and on their sides to advertise their distastefulness. But they're out during the day, so they're using their eyes during the day like we do. We can take those eyes, we can section them, and as I showed you before, and look at them. We can take really thin sections of the retina now, and you can see the photoreceptors out here. Here is it looks like a carpet almost out here of the rods, the outer segments, so we can measure their length and look at their diameter right there, and calculate their sensitivity based on their optical dimensions. So here are slices or examples, I should say, from our 4 species. Here are the 2 nocturnal on the left, the 2 diurnal on the right. I'll keep that theme going through several slides. Hylocenaria, tree frog. Here are the rods here. Rhinopippians. I actually removed the epithelial cells right there, as you can see the rods sticking out nicely. And here's the poison dart frogs, Oophagopamillia and Mantella viridis. And so what you're looking at now is this outer segment area, the most outer part of the retina. And we're going to measure the size of those rods only. I'm not going to do any cone measurements on this talk, rods only. So we're looking at an equation, the diameter and length. Close up so you can see how easy it is to make these measurements. That's the scale bars. We can easily apply these and digitally calculate their size and width. You can see a few little cones sitting in right there. They're tiny, but these are the rods. And so for the nocturnal species on the left, this is length on the y-axis and the species on the x-axis. This is the length of the rod's outer segment. You can see that the rod length is longer in the nocturnal animals than in the retina of the diurnal animals. It's shorter. For their diameter, this is diameter in microns on the y-axis, species on the x-axis. The diameter is wider for the nocturnal animals than in the diurnal animals. There's also a little bit of difference between these 2 diurnal animals. I'm not really going to talk much about that, but across the light niches, as they're called, or the light ecologies, the nocturnal have bigger dimensions than the diurnal ones. All right, what about some of our other factors that we're looking at in this equation? The aperture and then the focal distance right here. So for the aperture, these are some of the easiest measurements to make. You can dilate these animals' eyes and hold them in, this is an infrared photograph, so there's no light on them right now, which would cause their eye to constrict, even though we have atropine on their eyes, so it's keeping their eyes dilated. And then we can use a in-plane ruler, essentially, to then digitally calculate the diameter or size of the aperture of the eyes. Next, for focal distance, you can take sections of the eyes. These are frozen sections at their largest point, and you can then measure the focal distance from the center of the lens back to the retina. So what does that tell us? Let's look at aperture. Aperture length in millimeters here. This is a species again. Our nocturnal eyes, our nocturnal species, have much bigger, significantly bigger apertures than do the diurnal species. For focal distance, our nocturnal eyes have longer focal distances than do the diurnal species. So if you divide those 2, if any of you work with cameras, kind of know what this means. This is related to an f-stop, but most people don't have real cameras anymore. We're just using our phones. It shows that one of these species has a significantly larger ratio, which makes it, this is a nocturnal species, much more sensitive or causes the equation to increase much more than do the other animals. So let's put all these numbers, though, together to get some sensitivities, though. Now that we have the aperture, the diameter, the focal distance, and the length, we can put in a constant for this absorption constant of photons, and we end up with this sensitivity. And so this is just like this. You don't even have to know what this real y-axis is, other than to say that higher numbers are more sensitive eyes, and the nocturnal species here on the left have a more sensitive, have a higher sensitivity than do the diurnal species. So people have been doing research on this for years. They just take an eye, they harvest it from a specimen, and then they make these calculations, and you end up with these huge tables of sensitivity. By the way, our sensitivity is 0.133, so we're way down here in the diurnal range. We fit with this diurnal eye. We're nowhere near the sensitivity of these nocturnal animals. Who has the highest numbers? It's often these animals that are found at the bottom of the deep sea, these deep sea fish that people bring up, and they have giant eyes, giant eyes. Now that was optical sensitivity. It's a calculation based on anatomy. What about physiological sensitivity? Just what does the retina respond to? For this, you need to make an electrophysiological measurement. And these experiments, if any of you have ever done electrophysiology, you essentially hook up the nervous system to an amplifier and give it some stimulus or change its voltage and watch how it responds. For this, you can put an animal underneath a light, and this is really a cartoon. The dome that we have is here. It's made for these kind of experiments. And you can put little electrodes on the cornea of the frog's eyes, and they're kind of like little contact lenses with a wire in them. And then you can flash light on the eyes, and you can measure an electrophysiological response coming out from the eye. It's just, this is a gross recording like an EKG is for your heart. So even though you're not hooked up to the heart muscle, you can pick up the change in potential through the skin, through the chest, through the bone. An EEG on your head would be an electroencephalogram that would pick up the neural activity through the skin, bone, etc. But this is an ERG, an electroretinogram, which picks up the response of the retina. And what you see is this is time and this is voltage right here. You see, first you see an A wave, this hyperpolarization, followed by this large B wave. And the B wave is very big, and it's generated by a change in physiology of the bipolar cell. So light's coming in the retina like that. Here are our photoreceptors. The photoreceptors are generating the A wave, and the next synapse in this circuit going back into the brain is the bipolar cells, and they're generating the B wave. And so you can measure the amplitude of those and figure out what the threshold is, or the change in sensitivity is for different light flashes. So when you give these different light flashes, you would start out, this is time right here, and this is amplitude of a light flash. You'd give a very dim flash, the same dim flash. So the y-axis here is the amplitude or brightness, if you will. And you give 4 of those and you average them together. You can average the responses together to get the response for this level of light. And then you got to wait a little bit, and then you give a brighter flash, 4 of those, and then you wait a little longer, and then you keep giving brighter and brighter flashes. So the experimental protocol looks like this: light intensity here, and you keep increasing the light intensity. We go through 19 steps. Each one of those steps has 4 flashes of light. Why do we have this long delay between the flashes? It's because the eye doesn't reset very quickly. And we all know this because if you've ever had a camera flash in your face where you look at the sun, you get an after image of the light. And we don't want the next set of flashes to be, the responses to be reduced by that ongoing response to the previous set of stimuli, that after image effect. So what do the data look like? Here's some raw data up here: time on the x-axis, voltage on the y-axis. I'm just showing you the responses to 4 light flashes, 4 different intensities of light flashes. Here are nocturnals on the left, diurnals on the right. And so for a very low flash, you get this response. A little brighter, and brighter still, you get this response. And if you measure the amplitude from 0 up to the peak of the B wave, you can plot that on the y-axis here as a function of the light intensity. So this is on a logarithmic scale, the light intensity, and this is the relative amplitude of the B wave, this physiological response here. So as you increase light intensity, the amplitude gets bigger and it saturates eventually. So these are the curves from the data up here, 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 and here. And I'm only showing you 4 of the traces, and those traces match the points that match the color on these curves. These are called V-log I curves. You never hear that unless you run electroretinagrams, but it's basically the voltage as a function of the logarithmic of light intensity. When you put all those together and run it in many animals, that was just for one animal each to illustrate some raw data for you. We have the V-log I curves for our nocturnal species here and our diurnal species here. So this is light intensity on the x-axis and relative response, males on the top row, females on the bottom row. And what you might already be seeing is that the curves for the diurnal animals are shifted a little to the right. Look how this curve is staying low and then moves up here for these light intensities, whereas for Ranopippians or Hylicinaria, they're already taking off and responding at very low light intensities. And so that thing that your eye is telling you right now, there's a difference in these curves, these are shifted to the right, is telling you there's a difference in the threshold of light intensity required to elicit a response in these retina. And so we can take a threshold, and for this study, our operational definition for threshold is the light intensity required to elicit a 10% response, or light intensity required to elicit a B wave that is 10% the size of the maximum B wave. And so let's look at what thresholds look like between these species. So just pay attention to these black squares right here. And if you look, this is log, this is threshold on the y-axis here. So this is getting dimmer and dimmer in light down here. And these are our 4 species. What you see that when the background is dark, this is called scotopic sensitivity. When there's no light in the background of that dome and we just flash a light in the dark, that these nocturnal retina respond at a lower light level. This is dimmer light down here, and this is a log scale. So we're talking about more than 10 times, 1/10, more than 10 times less light, or 1/10 the light required to elicit a response. Now, when there's light on in the background, we call that photopic. This is like daylight in the background. There's very little difference between the eye's threshold for a response. So I'm not going to spend a lot of time on the photopic response, but I do want you to pay attention to the fact that we have thresholds that are low for the retina, responding here for the nocturnals, and higher thresholds for these diurnal animals. Okay, so now we've measured 2 types of threshold. We've measured an optical calculated threshold, and we've measured a physiological threshold. Let's plot them against each other to see if it makes sense, to see if one predicts the other. So now we have optical sensitivity here. You remember that weird unit I told you about? It's called microns per steradian. It's just how much of the visual field is projected onto a micron of retina. Here's going higher sensitivity going this direction, and here's our threshold. So more sensitive is low. And you can see by this line that runs through these that optical sensitivity does indeed predict retinal threshold or retinal physiological sensitivity. So light habitat affects thresholds, and those thresholds, the actual number of those thresholds or quantification of those thresholds are determined by the optics. So this was all done by students, and you know what the neat thing about this is that no one had ever done this across species. A lot of people had made measurements on thresholds and electroretinagrams, and then in different species measured the optical or anatomical measurements of the eyes, but never put them together in the same animals. And so this has turned into a nice result, verifying a conclusion that people kind of thought were true, but now we quantitatively say, yes, it is true that optical sensitivity does predict retinal physiological sensitivity. And the paper was published this past year in Journal of Comparative Neurology. There's Robert, Caitlin, Keith and Whitney. Robert is now an MD student at University of Alabama, Birmingham. Caitlin's at University of Texas. Keith is now a PhD student at Rutgers, and Whitney is here in my lab at LSU in New Orleans. So let's get back to this for a second and just look at these thresholds here, because this relationship now, we need to talk about something that goes to my second motivation, and that is that, I wanted to be able to change sensitivity. And if you have a pathology in the retina, it's going to cause the sensitivity to rise, right? The threshold's going to go up. You're going to be less sensitive to light. And we want a drug or a therapy that can push the threshold down, a more sensitive retina to lower light levels. So how are we going to do that? Well, there's no animal model right now for endocrine modulation. And when someone tells me that there's no animal model for something, the first thing I think about are frogs, and in particular this frog right here, which is a Tungara frog, which is named after the sound of its call. If you remember, that sound went Tungara. And by the way, if you work on an animal that sings, a requirement is you have to learn how to imitate it. So that's a joke, by the way. So the Tungara frog is this animal here, and it gives me a chance to mention Crow's principle. Crow was a Nobel laureate in like the 1920s, so over 100 years ago, and as part of his presentation and essay to get his Nobel Prize, he mentioned that for every question in biology, there is an appropriate animal to ask and answer that question. And so I found that Tungara frogs are really good at answering a lot of questions, although not as many people work on them. They should. So if you take a Tungara frog, we can make the measurements that we just made. We can dilate its eyes and measure the aperture, measure the focal distance, measure the length and width, the diameter of the outer segments of the rods. We can run electroretinograms like you see here and get a V-log I curve and then solve for its threshold. But what we need to do now is experimentally manipulate the conditions that I mentioned earlier that retina is in particular, its endocrine or hormonal condition. And the way we did that was we ran the same experiments we were just running with measuring ERGs, but in some animals, we took a subject and we either injected them with human chorionic gonadotropin, or we gave them no treatment, or just saline. And then you dark adapt them, you put them in the dark for 16 hours, and then you run their electroretinogram. You measure their physiological response of the retina. And again, HCG is a form of luteinizing hormone, which is part of the effect of increasing estrogen production. I can tell you when you do this to female frogs, you inject this, what do? After a few hours, they begin to ovulate. And so we know we have this independent confirmation that the hormone treatment is causing a reproductive state. Well, here are those V-log I curves. This is light intensity on the x-axis. This is the relative response. And we're looking at the B wave, the amplitude of the B wave. And the upper row is untreated individuals, and the bottom row here is individuals treated with human chorionic gonadotropin. So it's hard to see from these curves if there's any shift in threshold. So I'll show you them plotted on the same graph. So light intensity on the x-axis, relative B wave response. So this is a dimmer flash of light down here, and this is a brighter flash of light over here. If you look at the untreated animals, you can see there's no response for flashes at this really dim level, and then all of a sudden it begins to take off in these the filled black circles here as it moves up like that. Now for the group that has been injected with human chorionic gonadotropin, the luteinizing hormone, this curve starts earlier. You can see the fit to these data at this point is shifted to the left, but just look at any set of points. There's an untreated individual there, and then if you look at the red point, it's higher, meaning for the same level of light, there is an increase in physiological response. The nervous system is responding with a greater change in physiological potential, voltage potential, to the same stimulus of light. For this point right here, which matches this stimulus here, the same increase, there's really big differences here. The variance gets big in the middle of sigmoidal curves. That's part of what happens on these steep parts right here. But you can see even all the way up to where you get into saturation, we've got bigger responses. And so we've got a shift in the retinal sensitivity when these animals are in a reproductive state. We've got a shift in retinal sensitivity to dimmer light. Wow. That was really cool. Really, really cool. Because no one had shown that before. And what that means from a functional point of view in this animal's ecology is that likely for mate-searching individuals out in a very dark, on the forest floor, in the rainforest, and tundra frogs lived from about Veracruz, Mexico, down into northern Argentina, that in these dark habitats, any little bit of increase in sensitivity is likely going to enable them to more quickly or better find singing males on the forest floor. So I just showed you it on one graph. Here's another view of the same information. Here's the threshold right here for an untreated female. So this is this is threshold at which they respond. So this is dimmer and dimmer and dimmer as we go down like this. And this is for the treated female with human chorionic gonadotropin. And so we have this shift in threshold, and this is logarithmic units. So we've got over a 10-fold change in threshold, or it's at 1/10, actually more like in the 1/13th or 1/14th level of light required to elicit a response. Some of you are thinking, yeah, but what about the males? I didn't mention the males. There's no effect of this hormone treatment on the males. This is an untreated male, and this is a treated male. What does that mean? It means there's several ways you can look at this. One is that, well, we did the experiment right, and males don't have a hormonal mechanism for shifting the sensitivity. They could have a hormonal mechanism, but it doesn't run through a luteinizing hormone like we injected. Or there's some combination of the two and we didn't get it right. But because you can't explain these negative results very well, but this is conclusive that we have an effect of this endocrine state on the retinal sensitivity. So the male story is still open at this point. It's an open question. How does that map on to that relationship that I got so excited about that we published a paper on earlier, and that is optical sensitivity versus retinal threshold. Here are the data I showed you before. Optical sensitivity, more sensitive eye coming out here, lower threshold, meaning more sensitive eye going down on this axis for those other 4 species of frogs. When you put the tundra frogs, by the way, their species, genus and species name is Phycelamus pustulosus. So that's why you see PP here. When you put their sensitivity and threshold on this plot, they don't match the curve. And that kind of scared us a bit because we were really excited that we had locked down this relationship between the optical anatomy, if you will, and threshold. And so these were saying, no, they have this great optical anatomy for a very sensitive eye, but the retina is saying they're not using that sensitivity. But what happens when you put our endocrine shifted, our reproductive eye, if you will, on this curve? And the answer is that now we see that under hormone modulation, the females are now using all or the extent of their optical sensitivity. So this was exciting. This means that under non-reproductive conditions, these females at least are sitting in a relatively unsensitive or non-responsive retina. But when they become reproductive, the retina really makes use of the optical anatomy to become very sensitive. It suggests that there must be some cost to the retina to being in this condition all the time. It could be that the daytime blinds the retina, for example, or it could be energetically costly. Spend a lot of ATP keeping the retina in such a sensitive state, for example. That's still open. I'm just hand waving at what could be the explanation for that. As I mentioned before, the males, they never really get to their sensitivity, so the males are an open question right now. So, how to modulate sensitivity of a remaining retina? Right now, we're actively seeking, grant money. We're going to continue these studies both from a neuroethological point of view. I want to know about the biology of these eyes, but also thinking about we may be onto something that could be eventually a therapeutic tool for retina that have reduced sensitivity, and that was to make use of its endocrine mechanisms. Got ahead of myself, already mentioned that. Does this sensitivity have an effect on behavior? This was all physiologically measured. We were measuring. You can take these you can take these frogs and put them in a what's called an optokinetic response. And if you've ever had where something moves around you and it made you feel like you were moving and you kind of stabilized yourself, that's how you generate an optokinetic response, meaning the eyes generate movement, optokinetic. And so you can put these frogs in a little spinning wheel and see if they'll shift their weight to match the spin of the light that's going around them. And so the orange is the optokinetic response, meaning the threshold to elicit this behavioral response. These frogs are just behaving in a little arena with the light moving around them. And then the purple right here are the data I showed you before for thresholds for electroretinogram. So I'll cut to the chase, meaning that in a non-reproductive female, this is the threshold to elicit this behavioral response, meaning this is the light level spinning around their eyes that causes them to shift their weight in response to that visual stimulus, that moving visual stimulus. And this was the electroretinogram threshold. When you inject females, we get this lower retinal threshold, like I show you. But look here, the optokinetic response decreases at the same scale that the retinal threshold decreased. And so we consider this independent confirmation. These are done on different animals in a different set of tests. And we have the same scale of decrease in threshold, one a behavioral measure and the other a physiological measure, that are consistent, suggesting that indeed, when you change the threshold of a retina, you're going to change the behavior to light in the animal. The males, poor males, they just don't shift at all in this paradigm. We're still, like I said, they're still a mystery. This was published by some of our collaborators at Texas. Molly Cummings ran those experiments. So our data on this shift in retinal sensitivity, again, was published in Frontiers of Behavioral Neuroscience, and I'm really talking about that to get you all watching excited for what you can do really at this stage of your career by getting responsibilities in a lab, and hopefully we'll get to that point where we can get out of the pandemic. I'm really hopeful for that. Caitlin, Robert, and Whitney worked on this project. This is really their data. All of us at the end are really just guiding them in equipment use and data analysis, etc. But they were doing the hard work, and it's exciting to see them write this up and get it published. What's left to do? In the last just couple of minutes, I'll say quickly what's left to do. What's the mechanism of this modulation? Right now, the lab, I don't have a story to tell you other than the lab is looking at estrogen receptors in the retina. This is labeling, immunohistochemistry label of receptors in the retina, just showing you where the outer segments of the rods are here, and where cones are. These are the diurnals. Right there, they have more cones than the nocturnals do. They have more rods. But in these retina, if you look more closely for estrogen receptors, you find that the retina is just loaded with estrogen receptors. This is a G protein version of an estrogen receptor. It's on the membrane of the cell, but they have the nuclear receptors too, alpha and beta receptors for estrogen. So the world is wide open right now for how we're going to tackle this mechanism and really start to do reductionist sciences, reduce the unanswered questions down to a very fine point to figure out what the mechanism is. Just to orient you right here, here is a crescent violet stain of a retina. There's our outer segments right there. Light's coming from down here, remember. And here are the outer segments. These estrogen receptors are not found in the outer segments that are right in this layer right here, which is called the internuclear layer, which is what generates, if you remember, what generates the B wave, which is what we measured to be changing in response. So we're excited to look at estrogen receptor as a possible target for mediating, or at least being part of the mediation of this effect. What else is left to do? We were only presenting white light, but Whitney right now is doing these modulations, or looking for modulation for light of different wavelengths. And so on the y-axis here, this is the B wave size relative to a white light response. And when you inject human chorionic gonadotropin, we're getting these larger responses to particular wavelengths, especially red wavelengths and green wavelength. There's suggestion of change here for blue and orange, but it wasn't statistically significant. So that's where we are right now. That's where the lab is right now, going after the estrogen receptors and really untangling what parts of the white light is getting modulated by this endocrine mechanism in the retina. So I'll stop here and say that, and to sum up and say, I've characterized the optical morphology. We've characterized the optical morphology of these eyes. We've calculated from that morphology the optical sensitivity. Then we used electroretinograms to quantify the relationship between the physiological sensitivity and the optical predicted sensitivity. And we see that there's species differences in sensitivity, and they're really large under the scotopic conditions, meaning under nighttime conditions. But under daytime conditions, there wasn't that much of a difference in sensitivity. We've established that there's endocrine modulation in the outer areas of the retina, but we need to know what we still don't know what that mechanism is. We've shown that when this occurs, when this modulation occurs, it enables at least one species, the tumor frogs, to use the maximal amount of their optical sensitivity. And so we've starting to identify the endocrine targets, and we've begun to quantify stimulus specificity, with wavelengths of light, durations of light, speed, all that kind of stuff that are responsive to this modulation. So remember this slide earlier where I said I'm going to work on multi-sensory integration? That's what I'm interested in. I gave you this big talk of how great that is in a few slides. Well, we haven't gotten there yet because this is a lesson in following your data. But initially, when we just wanted to set these experiments up and get these animals in the right physiological context, we started learning that this physiological context was different than what anyone else was measuring in any system, not just frogs, but in any system. And so a take-home message here, besides learning about a neuroendocrine interface in the retina, is to follow your data. Let the data be the guide. Don't keep asking for different data just because you want a result. Let the data tell you what's going on. And in fact, there's an old saying, your data don't have to be what you want, they just have to be consistent. When the data start being consistent, they're leading you to a conclusion and can lead you down a really important path that can set up years of work, not just a couple of papers or a set of experiments, but years of work. And so that's where we're going right now. We're going to get to this integration at some point. I'm not the white-haired old man yet, teaching biology, but I'm getting there, and I want to do it, figure this out before I get there. But right now, we need to figure out what the neuroendocrine interface is in the retina, because I think it's very important for understanding the biology of the retina and for patient populations as well. So thanks to all these folks, Keith, Whitney, Robert, and Caitlin, and all our collaborators on this. Allison and Fern really helped in making our mentoring and pipeline programs possible to help me get folks in my lab who are awesome. And these are all our support down here. So I'll take any questions. Thank you very much.